Okay, I'd like to welcome everybody to the formal start of this year's CompuCell workshop. Depending on how you count, this is either the 17th, 18th, or 19th iteration of this. We appreciate people coming every year. We also understand that it's a lot of time and energy for you, and so we know that we do our best to try to make it interesting for you. Today, we will be introducing the basics of CompuCell. Some of this may be familiar from material that people saw during the orientation session, in which case it'll be a review. I need to remind you that, as always, the workshop is live streamed and recorded and made available on YouTube, so please be aware of that. We want to thank our funding agencies, the National Institutes of Health and the National Science Foundation, for their support of this project over the years. There's a Slack channel, which you can join, and Hayden will put the links in the chat. There's also the slide deck available in the student materials folder if you want to follow along. So today we're going to be focusing on a little bit of course logistics. Some of this will be repeating things that you might have heard a few minutes ago from Hayden, but some people are just joining in now. We'll invite you again to the hackathon if you'd like. We'll talk a tiny bit about virtual tissue models. We'll do more of that in the short session 1E a little bit later. And then we'll focus on running CompuCell. This will focus primarily on running CompuCell on nanoHUB because there are a few extra hoops to jump through when you're running on nanoHUB. Desktop people can try out running on nanoHUB as a backup. We recommend it. And then we will use CompuCell a little bit to begin to familiarize ourselves with how it works. And if we have time at the end, we will go very, very quickly through a little bit of how CompuCell specifies models. Again, all of this will be covered in much more detail later. The idea is to get a bit of a flavor for how things work so that when you see it the next time, it becomes more familiar. Logistics, again, audio is disabled by default. We will do a lot of polls. One thing about the polls, if there'll be a completion poll that says, are you done with this exercise? You can only answer it once. So if you're done, click yes. Only click need more time or not, please don't move on if we're about to do that. So the goal of this is to make sure that we don't move on before people are ready. But if you say click not ready 1 minute into the exercise, we don't have any way of knowing if you finished it 1 minute later. So there's a little bit of an inconvenience about the way Zoom polls work. Again, you've seen the list of people who very generously volunteered their time to work on this project. It's an entirely volunteer effort teaching this course. And we do appreciate all of the people who are helping out. The schedule is going to be 10 a.m. to 11:30 for Module 1, a short break, Module 2, an extra session, a short extra session. Each day will be different, lunch break, and then additional modules. There may be possibilities that some of the modules run over because the modules are Zoom streamed. We would rather have a module run a little bit long and change the timing slightly rather than cut modules up into different pieces from those that are advertised. Again, today you'll be getting CompuCell basics. You'll be hearing from me twice, and then you'll be hearing from Professor Giulio Belmonte from North Carolina State. We'll be introducing really the details of how CompuCell works. We have the hackathon coming up. We hope that people will participate. I'm going to do a very quick poll, just asking people what they're thinking about hackathon participation. If you say yes now, you don't have to do it. If you say no now, you can change your mind. But it is helpful to us to get a rough sense of how many people might like to participate in the hackathon. If you do want to participate in the hackathon, we put in the chat hackathon teaming documents. We will have more explanation of how this works tomorrow in the lunchtime session. But so if you would like to begin to look at what people's project ideas are, that would be great. If you don't have a project idea, but you would like suggestions, we could help with that as well. Again, if you want to propose an idea or see what ideas people have, there are Zoom links for that here, hackathon documents. You'll also have an opportunity to do a short one-slide talk on your idea to the rest of the participants to give build teams. Okay, we have a lot of people registered for this class. Not everybody who registers will come. We never know quite what the turnout is going to be. It looks like there are about 50 people in the room at the moment. We'll do our best to support each one of you, but please be patient. If you are an experienced CompuCell user, and there are a few experienced CompuCell users who are joining us and would like to help us out, helping people who are less experienced, please do let us know. Hayden is happy to talk to you about getting your help, especially for the hackathon. There are workshop resources. There's the CompuCell website, which has manuals on it. There are the class files. There's the Slack channel. We're going to be using next week a program called Antimony Tellurium. It's very simple programming language for describing reaction networks. There's a great manual for that online. And there are a variety of quick reference guides. The Slack, people should have seen. It works only if you use it. It's really a peer-to-peer communication vehicle, although we will monitor it as well. Please do take a look at it. The main quick reference guides, and we strongly recommend downloading them and printing them out. It's really helpful to have them on paper in front of you as well as online. There is a couple of pages on Python, the basics of Python. Really everything you need and more is in that Python sheet. If you look at the first page or two, you'll get 90% of what you'll need for CompuCell. Again, some people took the Python boot camp yesterday. Some people know Python well already. We will teach the Python concepts as we introduce them, but it's always good to have some backup for that. These days, I would also recommend, if you like it, using ChatGPT. In my experience, ChatGPT is actually pretty good at generating CompuCell Python code. It's worth a try. It may speed things up for you. If you discover good ways to use ChatGPT in the context of CompuCell, maybe you can come back next year and teach us how to do that. That would be great. There is a simple CompuCell quick guide called the Quick Reference Guide, which lists the key ideas in CompuCell and the commands. And then for network modeling, there's a one-page, two-sided Tellurium guide, which is also pretty useful. If you'd like to learn a little bit more about how CompuCell works and the backgrounds of it, there is a book chapter called Magnetization to Morphogenesis, which will teach you the ins and outs of CompuCell internals. That's primarily going to be useful for people who have some physics and mathematics background. You don't need to know that, but if you're interested, it's there. There's also a nice reference, not about CompuCell and not really about the kind of modeling we do, but for people who've never done any quantitative modeling, there was a beautiful paper published a few years ago by a group of Canadian students, graduate students, called 10 Simple Rules for Tackling Your First Mathematical Model. It's an open source paper, and I recommend it. Can't say I agree with everything in the paper. Not all of it is quite what we need for CompuCell. But if you've never done mathematical modeling before and you want to think about what you need to do to build meaningful mathematical models, it's a great place to start, and I strongly recommend reading it. Nobody comes to this kind of course with a complete background. Some people have more mathematics, some people more physics, some people more biology, some more engineering, some more computing. Depending on your background, you may want to fill in a whole variety of directions. We like the Rosalind exercises online, free for learning Python. network modeling, there is a great textbook by Herbert Sauro, which is not very expensive, and there are great online demos. For mathematical biology, I like Essential Mathematical Biology by Britton. Gilbert's Developmental Biology is a wonderful place to learn basic biology of the kind that's important in CompuCell. And you can buy a 20-year-old edition for $1 or $2 online, which covers most of what you'll need. Some of these things are also in Wikipedia. It's a little bit more sophisticated. The Physical Biology of the Cell by Phillips. It's a beautiful book. If you want an easier introduction to biophysics, Phil Nelson's Physical Models of Living Systems, very good. There isn't yet a textbook on CompuCell. The closest thing we have is about 10 years old by Shannon Preziosi, and it's worth looking at, although some of these things have changed over the past few years. I'm going to talk more later about motivation for multicellular modeling, but the world we're going to be living in is life. We started out developing CompuCell many years ago, more than 20 years ago, trying to understand processes in embryonic development, how a fertilized egg becomes an adult. It's an interesting question with living organisms, how they maintain themselves. There are no rulers or blueprints in biology. It's not so easy for a mouse to stay a mouse or a human to stay a human for its lifespan. Developmental diseases occur when the mechanisms of constructing an organism or maintaining it fail. The classic example of that is cancer, but there are many other developmental diseases. Everything from arthritis to Alzheimer's can be thought of in some ways as a developmental disease. Starting with COVID, a lot of people use this kind of modeling to understand infectious diseases and immune response, and that's a growing area of application. In bioengineering, we can use this kind of modeling to predict what would happen in a bioreactor or an organoid culture or a lab on a chip. So we don't have to model intact living organisms. We can also model living components in non-standard organizations. And something that's become quite important and a big application of this kind of modeling is toxicology, trying to understand how molecular level perturbations disrupt system level organization. That could be for anything from shampoo getting into your eye to a chemical disrupting your liver. And a lot of toxicological applications these days do use CompuCell. All of these require understanding a combination of physical and biochemical mechanisms. And these kinds of computational models are really important if you want to go from the parts lists that modern molecular biology gives you to a dynamic understanding of how organisms work. We often call these kinds of systems virtual tissues. What is a virtual tissue? Virtual tissue is a mechanistic, agent-based multi-scale model. Agents, that is, you have individual components that do things independently and interacting. The focus is on emergent spatiotemporal phenomena and on interactions between agents. And that's pretty important. We can't model all of the detail going on inside a single cell. And if the behavior of a tissue depended on all of that detail, we wouldn't be able to build computer models. These are mechanistic models as opposed to data-based models. They're additive. You start with nothing and you build. And that means you can't take anything for granted. You get nothing or almost nothing for free in a model of this kind. If you don't say that cells have a volume, they don't have a volume. So you have to remember that even the things that seem obvious aren't in this kind of modeling. And we'll build submodels of object behaviors, how cells are born, how they die, what forces they exert, how they change shape, how they move. And with CompuCell, you could typically model hundreds to hundreds of thousands of cells. So thinking about what you see in a 20x objective microscope about if you were doing an experiment. And the output will be movies. And one of the key things we'll have to think about is how to interpret those movies to give us meaningful information. Biology has a lot of length scales, everything from subatomic all the way up to whole body and population. It's highly heterogeneous. There's a lot of complex structure and organization at each scale, and there are complex interactions within and between scales. That makes it as complicated or more complicated than any other system in the universe. A mouse is a much more complicated organization than a 747. One of the things that's especially visible in early development, but occurs in all biology, is that you'll see a lot of movement. The top is an example of branching morphogenesis in vitro. Top right is a beautiful older movie by a group that imaged every cell in a developing zebrafish embryo. The spots are the nuclei of the embryo, and you can see how much dynamic movement is happening during the early morphogenesis of the embryo. You start out with everything dispersed, and then you can actually see the body axis there and the vertebra beginning to form. The bottom left is a live image of blood vessels growing, again in a zebrafish embryo. And then on the right is the early development of the body plan in the chick, again forming the body axis and the somites that are going to lead to the vertebra. So you see a hugely dynamic organization. That dynamics is less obvious in an intact adult, although there's some of that. Immune cells run around a lot. There's reorganization due to tissue damage. So the kinds of modeling that we're going to do are typically going to be focused on situations where there's a lot of cellular reorganization or a lot of cell birth and death. We're going to divide problems typically into 3 levels, sometimes 4. We're going to have cells and what they do, because cells are the primary active agent at the tissue level in organisms. We're going to have inside of them some concept of control, typically represented by networks representing gene regulatory and signaling networks inside the cell, sometimes metabolic networks. Those cells are going to interact with each other and with the extracellular environment, in particular by chemical fields that they secrete and that diffuse in the environment and that other cells can receive. Cells can change their behaviors, reorganize. In this case, is a simulation of that chick somatogenesis that we saw in the movie in the earlier slide. Pretty often, we will take a model of this kind and PK models, which represent the transport of chemicals primarily in the blood. If we want to know what happens when you take a pill and that chemical is redistributed in your body to your liver, to the target tissue, to your kidneys, to your brain, we can do that kind of modeling. And so we have these multiple scales. We have the cell, we have what's inside the cell, we have the local environment around the tissue, and then potentially we put that into a simulation of the whole organism. We could use these kinds of simulations to understand basic biology, to predict the effects of chemicals for good or bad, to design therapies, sometimes to personalize therapies for individuals, and particularly in the context of cancer treatment. And as I've said, these kinds of models that we're going to be focusing on the next 2 weeks are particularly useful when we have reorganization of tissues or movement of cells. There are other methods that are very useful dealing with very large-scale organization, which typically don't include cell movement or tissue reorganization. These kinds of virtual tissues can be very productive. You could use them as variable magnification microscopes to zoom in on problems. You can try to control different kinds of perturbations to understand when systems recover from perturbation or when the perturbation is amplified. Sometimes you can test hypotheses that are hard to test in experiments or infer parameters that are hard to measure. You can try out lots of different combinations that would be much too expensive to do in an experiment. They can be good communication vehicles because their movies are very immediately appealing and often make evident things that are not easy to understand in still images. They can be ways to test how much you know about a system and to determine what new experiments that you have to do. And it's a wonderful way of exploring new conditions. and possibilities that have never occurred in reality. They're not easy to build. You have lots of different components. Tissues have many more cells than we can simulate. There are many different types. Unlike, for example, in engineered systems, two cells of the same type aren't the same. And that heterogeneity, that variability is something that we have to deal with. There's a lot we don't know. We don't have design principle documents for organisms. It's often hard to compare experiments and simulation. And one of the things I hope people develop over the next 2 weeks is an understanding of the kinds of metrics, the kinds of measurements you could do on an experiment or a simulation to try to compare and understand whether the simulation and the experiment agree. There's also a lot of tight coupling. As I said, there's a lot of feedback in these systems, and that feedback makes both model description and simulation more difficult. We're also always in the realm of phenomenology. We're never going to be able to describe things perfectly. And so we have to be aware of the limitations of the models that we build. When we build models, there are many ways we could do it. We can represent cells as point particles. That lets us represent lots of cells at once. PhysiCell, which is also developed here at Indiana University by Paul Macklin's group, is a great example of that. Tissue Forge, developed by TJ Seago at University of Florida, is another great center model package. And there are plenty of others. Those are wonderful if you want to model lots and lots of cells, but the cells don't have any spatial detail. They're spheres or ellipses, for example. If we want to build models where the cells actually have shape, we can go into great detail. There's wonderful work, for example, by Karniadakis, who's using solids modelings and fluid modeling package to model the flow of blood. Those are very expensive computer simulations because of all that detail. And there are a whole variety of other methods that vary in the level of complexity and the level of biomechanics, the level of spatial detail that they offer. And we have to decide for each problem what level of detail we need to be able to solve the problem we care about. We're going to be using CompuCell3D. CompuCell3D will specify models using a combination of XML, CC3D ML, that is, and Python. It lets you define the layers that I've talked about of description. We'll be using the Twedit editor, which is specifically designed for CompuCell. It has, as a feature, a lot of the key Python and XML built in so that you can template off of that. So you don't have to remember what the command is to do this or that. It'll give you boilerplate code that you can then edit and specialize. And that makes it a lot easier to build. Many people that did it this year are running on desktop. But there's also that nanoHUB version, which I'll be showing you a little bit as well. CompuCell supports a number of concepts, and we'll see this slide again. The key concepts are fields, the idea of particularly a chemical field, although there could be other kinds of fields in our system, which essentially define the concentration of something, often a chemical species at each point. That field will typically diffuse in the environment and decay in the environment. It will be absorbed and secreted by cells and other components. There'll be cells and what are called generalized cells, which could represent, for example, components of cells like the nucleus. They'll have basic things like volume, surface area. They'll have more biological concepts, typically like polarity or planar polarity. And they'll do things like move, grow, decay, divide. They'll be able to stick to each other. They'll be able to respond to chemical fields. They'll change type. They'll secrete chemicals. They'll absorb chemicals. There's also a concept of links, which we'll get to next week. The cells that are basically defined in CompuCell are fluid-like. In biological systems, you have, for example, junctional adhesions that are more like staples. They're relatively permanent. You also have long fiber structures like filopodia, and links are a computational object that allows you to build representations of those kinds of structures in your simulation. And then importantly for the concept of control, you have networks Those could be gene regulatory networks, metabolic networks, or signaling networks. And those can be simulated in a variety of ways. There are a variety of languages for specifying them, SBML, Atomony, MabBoss, CellML. Conceptually, typically you either have what are called stochastic solvers, which could be Boolean, true or false stochastic solvers, or dynamic solvers like Gillespie algorithms. And you could also have reaction kinetic solvers, that is, modeling changes in state as ordinary differential equations. And next week, we'll go into how to do that in some detail. So the first thing I want to do before we take too much more time just talking is getting people to use CompuCell a little bit. And I want to start out by having people run a little angiogenesis simulation. This won't require you to upload or download anything. We're going to be building simulations in a little bit. At the moment, we're going to just run a simulation that comes prepackaged with CompuCell. CompuCell has a bunch of demos built in. The demos are not organized in the most friendly fashion, and they're not indexed very well. And so one of the challenges in CompuCell is finding a demo. If you want to learn how to do something, there's almost always a built-in demo with CompuCell that'll do it. Finding it may not be so easy. And we'd very much appreciate your help with our documentation and your suggestions for ways to make those demos a little bit more usable. We're going to be talking about vasculogenesis and angiogenesis. This is how blood vessels organize, both in early development and later on in life. And it's based on a series of simulations that were done by Roland Merks, who was in those days in my lab, and is now a professor in Leiden University in the Netherlands. In this case, you have a set of cells called vascular endothelial cells. Those vascular endothelial cells interact with each other to form a pattern that looks a little bit like chicken wire, dynamic chicken wire, of struts. Later on, those struts will hollow out actually to form blood vessels. You can also have existing blood vessels that sprout. And one of the questions that we were interested in the old days was whether the same mechanism could explain both self-organization of blood vessels by dispersed cells and sprouting angiogenesis. There were some interesting experiments and simulations that suggested that there was a very simple mechanism at work here, that the cell secreted a chemical that diffused in a short range in its environment. Chemical is called vascular endothelial growth factor A, VEGF-A. If people have studied cancer, they'll have seen that name a lot because it's associated with the growth of blood vessels in tumors as well. And that the cells move up gradients of that chemical. There were also experiments that we were connected to by our Kansas collaborators that showed that when two cells, vascular endothelial cells, were touching each other, the surfaces where they touched didn't respond to the external chemical. That's called contact inhibition of chemotaxis. And we did some experiments that showed that that was the case. Specifically, if you knocked out that contact inhibition, you didn't get vascular patterning. And so we built a simple simulation of that process. Here you see the cells. The background color is the concentration of the vascular endothelial growth factor field that the cells are secreting. On the right, the cells are not responding to the chemical where they touch each other. On the left, they are. On the left, you see that they form blobs. On the right, they form a vascular pattern. On the top is an in vitro experiment by Ambrosi et al., where they took human umbilical vein endothelial cells, put them in a culture dish with particular conditions, and you see those cells forming a vascular pattern. They start out dispersed, they come together to form aggregates, they form struts, and they gradually organize to form a vascular pattern. Let's run a little simulation of that together. Later in the course, you're going to understand a lot more about the background of this, the physics of it, the biology, and also you'll build the simulation yourselves. Today, we're just going to run it. If you're running this on desktop, you can do this in only one step. For nanoHUB, you have a couple of extra steps. We recommend you try the nanoHUB anyway, even if you're going to run desktop, just to see how it goes. Remember that the nanoHUB deployment is CC3D base 4X. If somebody could put that link in the chat for everybody, that would be helpful. Remember, go to nanoHUB and log in, and I'm going to do a split screen so we can do this together. Okay, so go to nanoHUB. Come back here for a second. Go to nanoHUB, log in using your preferred method. If anybody is not into nanoHUB, let me know. I'll do a quick poll just to make sure that everybody is able to get into nanoHUB. Okay, once you're in, you'll need to be able to get to your dashboard. Here, I've got the dashboard already. If I go to menu, I can select dashboard here. Or if I click up on nanoHUB on the top left, I can also select dashboard. Okay, once I'm in the dashboard, I am going to look for CompuCell. I can go to all tools here, search. I can type CompuCell3D, and I can do a search. And I'll see a bunch of CompuCell options. And the one I want is called CompuCell3D V4 main tool. It may be better actually if I type V4 here to make sure that I search it properly. Everybody able to do that? Once you've found it, hit this little heart to bookmark it. Then you won't have to keep looking for it again and again. If you search here, you can go, once you're logged in, you can go directly to this site. Like that. And so I want to make sure everybody has found that. Once you've found it, start it by hitting Launch Tool here. Or if I were in the previous screen, I can click on this button here, this little screen button to launch CompuCell. So please, everybody, launch CompuCell with me. It may take a second to get started. Does that display properly for everybody? Again, I'll do a quick poll. I won't be doing polls this much going forward, but to start out, I want to make sure that everybody's able to keep up. If you're running on desktop, launch Player. Launch Twedit. We could launch Player too. CompuCell comes with a number of demos. You can see actually from the title that's displayed in the desktop box here that it's already running angiogenesis. We're going to open the angiogenesis simulation here. Again, you have two tools here. One called Player. You see CC3D Player up at the top. On desktop, these will be separate applications. In nanoHUB, they're actually packaged together where you switch with tabs down here at the bottom. Okay. On desktop, you manipulate Windows the way you would in any other application. In nanoHUB, you can pull and resize windows inside the bounding box. The other thing is you may need to be able to resize your bounding box window like that. One thing that you may run into in nanoHUB is that the windows don't redraw properly. If you resize the bounding box by pulling on that bottom right corner, it forces a redraw. So if you have a screen that looks messed up, you pull on that bottom right-hand corner, that'll clean up the screen. That's a nanoHUB specific glitch. Okay. And you can resize the components inside your simulation as well. I'll show you how to get to the angiogenesis model in both desktop and nanoHUB just a second. Okay. So, as I mentioned, CompuCell is pretty good about hiding its demos. It has great demos, but it's a little bit hard to find them. Let's find it together. Go to the file here. Go to Open Simulation File. If you're on nanoHUB, you select your name here, whatever your name is for your account. If you are on desktop, you need to find the directory in which CompuCell is stored. Open under your name. And you will see I have a lot of things in my nanoHUB account because I've been using it for a long time. But you will see a folder called CompuCell3D Demos. And on desktop, you'll find the same folder. Everybody make sure they find CompuCell3D Demos. On desktop, wherever you find that, it's okay. On nanoHUB, it's important that you first select your username. There's going to be a competing version of CompuCell3D Demos under computer, which is not writable. And if you try to open that one, you'll get an error. If it says slime mold, that's okay. Somebody asked, how do I find slime mold? Go back to open the file here and go to demos. Question, how do we do this on desktop? On desktop, you open up Player, you select File, and then you navigate to wherever you install CompuCell. In the CompuCell directory, you will find CompuCell3D _demos. Okay, open CC3D demos here. You'll get a whole bunch. You see slime mold, patch, lots of them here. Yes, I apologize. I had things reorganized in a funny way for myself. So it shows you that the organization of the demos really can confuse somebody who uses them all the time. Sort by name. There'll be a subdirectory called demos inside of CompuCell3D demos. Demos. Then we select book chapters demos. And then we select angiogenesis. That rather long path name is here on the screen. CompuCell3D _demos, demos, book chapter demos, angiogenesis. Somebody says I do not have demos inside of demos. Do you see book chapter demos _ computational methods? There may have been a reorganization in the last release. And so nanoHUB may have a slightly different architecture from the demos on the desktop. Sharon, can you find it? Book chapter you found. Okay, if you could put the path name in the chat for the desktop people, that would be extremely helpful.
Yes, on desktop, when you go to open, it should say, sorry, let me pull it up here. This PC, Windows, CompuCell3D, there's just a folder called demos. You won't have the nested demos folder.
That's actually a fix. I mean, that was something that we wanted to correct. But the problem is that the version of CompuCell on nanoHUB is one version out older than the one that you're using on desktop. Everybody okay with that now? Looks like everybody's got it. Select Angiogenesis CC3D here. Hit open. And you should see nothing. And now I can hit the play button. And we can begin to use Player. So let me give you a little bit of guidance here before I get too far along here. You'll see at the top left of your menu bar, and this will be true in desktop or in nanoHUB, 4 buttons. A square button that's labeled stop, which you don't use unless you really want to terminate a simulation, because you have to restart the simulation from the beginning after using that. A circle button that looks like a record button in an old cassette recorder player that single steps the simulation. An arrow button, play button that's run. And when you're running, a blue pause button, which you'll use a lot. So try that out. Try hitting play, pause, step. I don't recommend hitting stop because you have to restart the simulation. If I hit stop, the simulation will terminate. And then if I hit play again, it will start again from the beginning, and I'll have to resize my screen. People should try that out a little bit. I'll leave it running while we're doing that. You may see something that looks a little bit different. There are a variety of configuration options that you'll have. Something that's very useful is if I go under Window here and I select Tile, it will lay out the screen appropriately. That's one of the most useful functions we have. That works on Windows and it works on nanoHUB. It will not work on a Mac. The Mac windowing system does not allow that functionality. So I'm afraid the Mac people have to lay out windows by hand. But the Windows people and the nanoHUB people get that benefit. You'll have a variety of selection options. You can view the simulation in a 2D slice like this, or in 3D. I recommend everybody switch to 3D here. Pull down here, 3D. When you change your viewing option, the system will pause. And so if I want to keep running it, I have to hit run again. Okay. Any questions about that so far? You'll have plenty of experience with this as we go forward. I can do a resize if I need to run my screen on nanoHUB, and I can change the size of my domains. Okay. So why don't people try playing with that a little bit, run the simulation, practice switching from 2D to 3D, practice what you can do. You can scroll, rotate, resize. If you make a mess of your window, there's a function called reset, which is very convenient, which is hover over the window and hit the letter R, and that will restore the view to its default. So if you say, somehow I managed to scroll off of the field of view, I don't know where my cells are anymore, hit R, and you'll be able to recover. Why don't people play with that just for a minute? Give people a chance to catch up. Everybody got a working version of this one way or the other on desktop or nanoHUB? Okay. A few people are asking for help. Please chat with Julio or Hayden for help. There's a comment that reset doesn't seem to do much on Macs. I'm afraid that the Mac people may have a little bit more trouble. You'll have to do a little bit more by hand. Somebody says the colors are different. So one of the things that we can do is change how we visualize in CompuCell. If we go to visualization, we have a variety of options. We can zoom in. We can zoom out. We can try reset graphics window. I don't know whether that works better on your Mac than the other R option. The other thing that we can do is turn on how we visualize. We can turn the cells off entirely. We don't usually want to do that. We can change the rendering of the cell borders, cluster borders we're not using at the moment, and so on. Those options are ones that we will use from time to time. Again, whenever we change those things, we'll have to hit play to resume the simulation. If you're on a slower computer, you may want to hit pause before you start manipulating the visualization, or you may get some delays. You hit pause, change the visualization, and then continue. Something that is very useful that we'll do a lot is use this configuration menu. We can find that in two places. This little screwdriver and wrench symbol here will give us configuration, or under tools, we can pull down configuration. We'll open that menu, and we'll be able to change the color of our cells. Somebody said their cells were a different color. I can change my cell color by clicking here, selecting the color that I want. Not a very pretty one there. And I can make that change. There are a variety of other changes that you're going to be making. If I want to save simulation output, that won't work very well on nanoHUB. It'll work great on desktop. I can change the cell colors, the border colors. When we come to chemical fields, I can view them as well. And so people could play with that a little bit. Somebody says that there was a view switching error in CompuCell. That one is a little bit tricky for me. I don't know. I'd have to look at your system. There's a question about Mac virtual environment. That one I can't help with either. You'll have to reach out to the assistants here to help out on. One of the reasons that we used to teach this course entirely on nanoHUB was that everybody had the same interface. One of the problems is that when people run desktop, is that depending on the specific computer and the specific installation that you're running, it's going to be a little bit different. And it makes it a little bit tough for us to debug everybody at the same time. It's not that we don't want to help out, and we're definitely willing to help out, but it's hard to anticipate the individual problems. There are people in the room who know how to help with all of these things, so definitely reach out for them. Okay. One thing that you may find is that you need to have a simulation loaded and at least stepped once before you can do any of these changes. And so you may find that if you start the simulation, if I hit stop, I can't make changes to the state until I hit step or run. You can try that out if you like. Okay. So the people who are running desktop and who are having problems can interact with the teaching assistants through the chat while I show people who are on nanoHUB how to upload and download files, because this is something that the desktop people will not have to worry about, and the nanoHUB people do. So if you're on desktop and you want to try nanoHUB, that's great. If you're on desktop and you want to debug or play more with the simulation that's running now, that's fine too. This will only take a couple of minutes. It's an inconvenience, and it's the cost of running on a public server. nanoHUB has some limitations on what you can upload and download, and one of those limitations is it does not allow CompuCell directly to upload or download files. And that means that we're going to have to upload and download files in a different way. The other thing that we can't do in nanoHUB, which is a bit of an annoyance, is that we cannot cut and paste between the model editor and other programs. You can cut and paste within the model editor, within Twedit, but you can't, for example, cut and paste from a Zoom chat into the model editor. And that's a limitation of the X11 protocol that's used in nanoHUB. And unfortunately, there isn't really anything we can do about that. We recommend that you use Jupyter Notebooks running in CompuCell as a backup that allows cut and paste. Again, if you're using desktop CompuCell, you can skip these steps. Okay, so that we're now going to go to nanoHUB and open Jupyter Notebooks, and that's what we're going to use to do uploads and downloads. So I'm going to go back to my dashboard. Everybody go back to your dashboard. If it will. Let's see. Okay, took a second, but I'm back to the dashboard. And now I would like people to search. You can search all here. Type in Jupyter. nanoHUB is a bit slow at the moment, probably because so many people are logged into it. Type in Jupyter, and what we want to do is find Jupyter Notebooks, and we want to find the latest version that we can. And the latest version, I think, is going to be 2021-05. And bookmark that. Okay, and then hit the little square button here to launch it. This is going to take a little bit of time to explain. Once you've done it a few times, it only takes a minute. The first time you do it, it's a little bit of an inconvenience. I'll put the link in the chat. Any one of these Jupyter Notebooks will work. Some of the older ones will give you a warning that you're using an older version, but they're all equivalent. Doesn't matter which one. Oh, sorry, I said just, I clicked on Jupyter 2019 when I should have clicked on 2021. Yes, thank you. Let me fix that. They all work the same. Yeah, you see it's giving me a message that I should use a newer version. Okay, let me put the newer version in the chat. Okay, launch the new one. Sometimes with nanoHUB, it'll give you a warning that you have too many versions running. In that case, you can kill the ones that you don't want. So let me come back and I'll put in the one that I want. Launch. And I'll make sure everybody's able to do that. All right. You see, I have a lot of things in my directory, so I'm going to have a bunch of stuff. Okay. To upload a file, we go into Jupyter Notebooks and we hit upload here. And within this, it will then point to a directory in your desktop. In this case, I want to go to Student Materials, Demo Code, Module 1.1, and I'm going to select the zipped version of Colonic Crypt. And everybody should have access to this file. It's been emailed to you. You can put it on your desktop, your downloads folder, anywhere that you can find it. I want you to find that and point to that file. Make sure everybody's able to find it. You have a direct link to it. The file is in Student Materials Demo Code Module 1.1. Somebody says they don't have the folder. The link is provided to you. No, don't unpack it. Leave it zipped. That's right. It's a zipped compressed folder. Okay. Let me explain what's going on here. CompuCell simulation specifications are multiple files. Having to upload a whole file structure is a pain. And so we typically keep them zipped so that you can deal with a single file at a time instead of having to upload and download multiple files together. Okay, let's try that again, see where we are. Again, once you've done this once or twice, it won't seem so difficult. I want to make sure people are able to get it to work. Again, you'll have to download from the Google Drive to your local machine. Put it anywhere you want. So Macs are being unhelpfully trying to do what they think you want to do. So yes, you will have to rezip it if the Mac has unzipped it when you download. If you're running desktop, it's not an issue. If you're running on nanoHUB, you need it zipped. Okay. Okay, select the example Colonic Crypt zipped, open, and then hit the blue upload button. In my case, I've already done this, and so if I want to do it again, I would hit overwrite. I'll do that. And it will now upload that file. Again, if this is not working well for you, you can practice during the break. We can go over it with you during a break, and you'll have lots of opportunity to practice this going forward. Once you have done this, we are going to go back to CompuCell. And we're going to go back to CompuCell by going back to our desktop. You'll see CompuCell here, and we'll select Open. So that's something you want to get used to, is going back and forth in nanoHUB between Jupyter Notebooks and CompuCell. Somebody is saying that the current version of CompuCell may crash when you use the configuration setting. We'll see about a workaround for that in the break. But in the moment, I guess don't use the configuration setting to change the settings for the moment. We'll do our best to get that fixed for you. Should be okay on nanoHUB. This is, again, one reason that nanoHUB, despite its inconveniences, is worth dealing with, which is that it has a uniform experience for everyone. Okay, come back to CompuCell3D. And now we're going to open that simulation that we just uploaded in Twedit. And to go to Twedit, we have a number of options. We can go under File and say Start Twedit here. We can go under Tool, here in this, I apologize there. We can go in this menu and you see this piece of paper and the pen, that's Twedit. So we can say Start Twedit. And you'll get an error message, which we'll ignore. Another error message, which we'll ignore. And you will see an editor window. So I want to make sure everybody's able to get to the editor. If you're on the desktop, just launch Twedit. Absolutely, the nanoHUB version may be slightly less powerful, but for the purpose of learning, we really encourage you to use the nanoHUB version. If you're having issues with desktop, use nanoHUB. For the next few days, none of the things that you're going to do are going to be that demanding. And then that we can give you time to get you support to get the desktop things working the way you need to offline, not during the middle of the course.
Yeah, I want to break it. Excuse me, sorry, break in here for a second and just say that the nanoHUB and desktop versions are going to be almost identical. So anything you learn through nanoHUB, you will be able to port over to desktop. You were looking at basically the same interface. So don't worry too much about, quote unquote, learning on nanoHUB. You're using the same software.
Yes. The only differences are there are going to be a few features that are not available in nanoHUB that are available in desktop, and you have to upload and download things. But 95% of it will be the same.
James, you may want to show how to resize the window. We have somebody, Edgar is saying that it got auto resized for him. You don't have to restart.
So if you want to resize your window, you have two things. First, the bounding box. You pick on the bottom right. You can pull on the bounding box and resize the bounding box. And then within that bounding box, I can resize the CompuCell viewing window here using the standard restore full screen tools, or I can pull on the bottom corner of the window as well. I need to switch within the nanoHUB application. I'll see tabs. I'll see a tab called Twedit, which is my editor tab, and a tab which is Player. And I can switch back and forth between those two. For the moment, go to Twedit, hit this up arrow button, which is Open Project, or you can do CC3D Project, Open Project, either one. Those are both possible. We select Open Project. And in this case, actually, it's already in the Colonic Crypt simulation that I've already unzipped, but I'm going to show you how to unzip. So you should see something like this. Point to your user directory. Find the file that you just uploaded. And I have a lot of them here. You should have much fewer things there. And you will see something which should say example colonic crypt.zip. Select that. And hit open. Find example colonic crypt.zip in your user directory, hit open. And it'll say something like could not create default unzip folder. It's okay. Hit okay. Save it. And just hit yes, ignore the warnings. Okay, everybody able to do that? That will unzip the file. Once you've unzipped it, I select open again. And now it should point to the inside of Colonic Crypt, and you should have selected highlighted Colonic Crypt.cc3D. If you're not seeing that, if you're going higher up, Here's the root directory. I scroll down and I should see Colonic Crypt. And I've numbered them because I had done this so many times. Here it is. Example Colonic Crypt. It's this one that I just uploaded. And I have to select open it inside of that because I have multiple copies. You should see multiple copies. Okay, people able to do that? Okay. Again, what you need to do is hit open. First, unzip the file, ignore the warnings, then hit open again, find the now unzipped file, and navigate to that. Okay, we'll give people just 1 or 2 more minutes to try to do that, figure that out. Once you've done it once, it'll work easily. Okay. Select colonic crypt.cc3D, hit open, and you'll see a window that looks like this. This is actually now, double click on the name. This is actually the simulation code that specifies the simulation. We're not going to look at that right at the moment, but that's what the simulation is. Once we have it loaded, you can hit the CompuCell icon here. You can hit under CC3D project, open in Player here. And we'll hit open in Player. Okay, we'll get a Player window here. And I'm going to resize this. And you should get something that looks like that. Any questions about that? Maybe instead of doing another poll, because I know the polls will interrupt what you see, if you could raise your hand if you need help or go into the chat. Everybody got that working? This is the simulation running. Okay. So we've got open in Player. Remember to switch back and forth. In this case, Player is actually launched twice. So I have 2 Player windows open. And I have tabs to switch between. I'm going to go to a 2D view here. And you should see something that looks like that. Again, to switch from 2D to 3D, I pull down 3D to XY. Okay. You can close the angiogenesis window, but you don't need to. And I realize the first time you do this, it's painful. Once you've done it, it takes about 60 seconds to 90 seconds to do. You can use this full screen button here, restore button, to resize your windows. Okay. All right. Keep working on this, keep practicing. You'll have lots of opportunity to practice again. Somebody says nanoHUB crashed. That's unusual. We'll see what we can do about that in a little bit later. If nanoHUB crashes, you can restart it. So to restart a nanoHUB application, you hit terminate here. You hit terminate and then relaunch it. Terminate. Then I'm going to relaunch CompuCell. Now I'm going to reopen the simulation. I can do recent simulations. It'll remember the last one that I ran. Remember that tile function is very helpful. And now I'm going to switch to 2D view. Something else that you might want to do is to download things from nanoHUB. This will typically be something that you'll do in Twedit rather than from Player. I'll start Twedit again. If I go to Twedit, and again, you'll have to ignore a few error messages here. I'm going to open that Colonic Crypt simulation again. And if I go into Twedit, I can go to CC3D project. I can select zip it, and that will package whatever I have within CompuCell file, the open CompuCell file, as a zip file. I've already saved this before. I'm not going to save it again, but you could say you'd hit save. And then you would do the same process that we did before, which is you go back to your Jupyter Notebook window. I go back to my Jupyter Notebook window here. I select the zipped file that I want to download. And then I hit download. When I do that, it will download it to my downloads folder. And so you may want to try that. Okay, so you can try that out. Since we're running out of time, we'll skip that now, but you'll have plenty of opportunity to practice. Something that I did want to bring up now, because we're going to run out of time, is that if you are, especially on a desktop, having player crash repeatedly, or a particular simulation crashes, that typically is happening because the settings configuration for player are corrupt. And the problem that we're going to face with that is that it's a hidden file. It's a file called settings.sqlite that has become corrupt. If you are having repeated crashes, what you need to do is go inside of the simulation directory and look for the file _settings.sqlite and delete it. If you delete that, you should find that you're able to run the simulation. If this happens repeatedly for all simulations, not just for one simulation, there's an equivalent version of that simulation settings file, settings.sqlite, that applies to CompuCell as a whole. You have to navigate to users, your username CompuCell3D_py. It's also hidden. Notice the dot at the front. And delete that SQLite file. And those instructions are going to be available to you to try out as you go forward. That's probably your best bet. People can use CompuCell for years and never have a problem with that SQLite file. But once you have a problem, if you don't know how to get rid of it, it's really rather annoying. The other thing that you should know is that if you use CompuCell with multiple monitors, the way I am today, and then you switch to using a single monitor configuration. So I'm talking to you from a laptop with an external monitor. I have CompuCell open on the external monitor. If I now close CompuCell and disconnect the external monitor, CompuCell will remember where the Player and Twedit should go. When you reopen CompuCell, you won't see anything because it will be opening them outside of the field of view of your current monitor. The easiest thing to do is to reconnect an external monitor, drag the CompuCell windows to your main window, and then close it. That should fix the problem. There is a workaround, and Giuliano is going to create a set of instructions for how to find that hidden screen. It's different on a Mac and Windows and Linux, which is why we don't have a single set of instructions at the moment. I don't think anybody's run into that particular problem. I've done it occasionally because I prepared for a class using 2 monitors, and I've gone into a classroom with a single monitor and said, where did my Twedit window go? So that's something that you may want to follow as well. Okay, I don't think we're going to have time to do the next exercise. We're out of time for this morning, which was going to be reading and writing and running a little CC3D simulation together. You'll be doing that in the next exercise anyway. This was a bit of a dry run, but there's no problem skipping that for today. If people want to see how to do it, the slide deck is available to you. I appreciate people's patience. There's always some teething problem at the beginning. In my experience, generally, there's a little less teething problem with nanoHUB than with the desktop, but there are issues with both. Once you get it running, it usually works pretty well.